Does this count as genetic modification? What's this a picture of? What's it a picture of the process of, from left to right? Selective breeding. This is the domestication of corn, maize.
I mean, this was done before anybody knew anything about it.
This is thousands of years old. Genetic modification, if you like that definition of genetic modification. This is breeding, taking existing mutants and turning them into something that's useful to eat. So thousands of years ago, and teosinte still exists, but thousands of years ago in central or southern Mexico, people started domesticating teosinte. They started selecting for the fruit that had certain desirable benefits. So here's a picture of a current teosinte plant. It doesn't look very much like corn. It looks kind of like a corn plant. And the domestication, as I said, occurred here, southern Mexico. There was a paper published recently that showed some evidence for this. They could actually go into a cave, a shelter, where maize used to be milled or ground by hand. And they found these rock tools they could extract particles of teosinte from. And the biological material they found embedded in these rocks looks more like teosinte than modern corn. So this is how they knew that at the time, and so they could date the age of these rocks, and that's how these scientists determined that it was literally thousands of years ago that this process was occurring, where they were domesticating and grinding the fruits of these plants and using them for food. So almost 10,000 years ago. So it's been a relatively long period of time since maize has been domesticated from teosinte. There's one critical difference between teosinte and maize. So this is just one publication out of many, and certainly this is only one step in a series of steps that leads from what teosinte is to what corn today looks like. But a key change from this paper in 2004, one nucleotide mutation, single nucleotide polymorphism, one SNP, changes teosinte, which has its fruits totally enclosed by this really hard I don't know what it is. I'm not a plant anatomist. Maybe somebody here can tell me. It's a gloom, but it's a really hard covering around the fruit. It makes it really hard for humans to get in and extract the fruit and use it for food. But there was a single mutation that opened up those glooms, the fruit cups, so you can maybe see it better on the right here in panel C, or you can start to see those fruits are a little bit exposed. They're not completely covered by the gloom. So now it's your turn. Oh my goodness, what am I trying to show you? Check it out. They've got DNA sequence from a particular gene. I'm circling in blue here on the left all of the maize plants. So what they did was they sequenced DNA from a number of different corn plants, modern corn, maize. And then in red, they got the sequence of the same genes from a bunch of different teosinte plants. So this is a phenotypic difference, blue versus red. Red is plants that have closed fruit cups, and blue open. So the question to you is, can you find a genetic difference in the gene sequences in the top panel, the blue versus the red, that explains this difference in the phenotype, open versus closed? The way to read this, just briefly, this top line is the actual DNA sequence. Underneath it, every dot, that just means it's the same nucleotide as the sequence above it. So they just didn't want to write out the same DNA sequence time after time after time. So the only DNA nucleotides you see down here are where there are changes, mutations compared to the top line. So before I let you go to work on this for a couple minutes, what's the big difference between the maize plants and the teosinte plants? No, big picture. Don't even look at letters. What's the big difference between the top half and the bottom half? The top plants, all of the maize plants, are very genetically similar to each other. There are very few letters written in here. That means, for example, this bottom row, MH16, you read across, it's got exactly the same sequence as the plant on the top, except for there's one mutation there. So all of our corn plants are very genetically similar to each other. All of the teosinte plants are very genetically different. Okay? So look through this table, see if you can find one nucleotide, one site, one column left to right that controls whether or not you've got open or closed fruit cups. Go for it, collaborate if you want, and in 2 minutes I'll reveal the answer. Dun, dun, dun. Spot the mutation that causes open fruit cups. Okay, so what's the magic mutation?
The very last one.
The very last column. So if you were looking from left to right, you may still be scanning across to figure this out. That's fine. So what is it about the very right column here?
Well, you have all guanine in any of the teosinte that's closed, and for some magic reason, as soon as you hit the open maize, it switches.
Right. So every single plant that is a teosinte plant that has a closed fruit cup has a guanine there. And every single plant that's a teosinte, maize has open, has a C. So every one of these is a C. That's what those dots mean. But then when we get down here to the bottom, where we're looking at teosinte plants, not a single one of them has a C. They've all got Gs. So that's a perfect, as we've seen before in class, the last time we did an example like this, it was with a pedigree. But this is the idea of looking for a correlation between a genotype and a phenotype. And that's how we find mutations that affect phenotypes. There's a perfect correlation between the nucleotide, and by the way, what number is that up there at the very top?
18.
This is nucleotide 18. What does that tell us? What are all of the other nucleotides up there? The numbers. What's the pattern? We've got 18 on the right. What happens as you move to the left? With the numbers.
Bigger?
They get bigger and they get, well, they don't get bigger.
Negative.
They get negative. So you should all know what that means. What are the negative numbers? They're upstream of the transcription start site. They're in the promoter. This one nucleotide, the very right side, number 18, what does it mean to us that it's a positive number? It's after the transcription start site. That is, it's the gene itself. So here it is. Position 18, right there. Teosinte has a G. This is what we just saw. Maize has a C. And in this publication from 2004, John Dobley reviews that causes a single amino acid change. One point mutation changes one amino acid in this protein from the teosinte gloom architecture gene, TGA. One mutation, one amino acid change makes that difference. Closed fruit cup, open fruit cup. Something that you can't use as food, something that you can use as food. Single mutation. I'm going to leave it to you to figure out, if you want, it's the G to C change at position 18 that changes a lysine, that's K, to asparagine, N. So the reason I put the codon table here is I'd like you to figure out what's the reading frame. Is it 16 through 18, is it 17 through 19, or is it 18 through 20? Fill in some of these nucleotides to figure out how a G to C mutation could change a lysine to an asparagine. It's just for practice. But a single point mutation that happened in nature at random allowed domestication. We could do this by genetic engineering today. Single point mutation. We could do that with CRISPR, probably. We could make a transgenic plant that makes exactly the same one change in the DNA. But that would be genetic modification and presumably would have no difference